So unlike 2020, which is a horrible year, 2014 actually was a quite nice year, especially when you're into avian genomics or avian research in general, because for those people who don't know, it was actually the year when a special issue in Science was released in December, led by these three gentlemen here, that compromised the release of more than 48 bird genomes. And that was surrounded in the Avian Phylogenomics project. That wasn't only 48 bird genomes that were released, but there were also associated papers, more than 25 papers, some of them really high impact. So for avian research and avian genomics, it was a really good year. So what made this possible? Well, probably all heard about DNA sequencing and its relation to the Moore's law. So what happened is, due to technological advances, DNA sequencing became not only cheaper, but became massively cheaper. In particular, there are two jumps. So the second generation sequencing here, close to 2008, and the third generation sequencing, close to 2015, basically led to a massive reduction. And suddenly, people started to sequence not only things that are closely related to humans or model organisms, but they could start to sequence genomes that are basically limited by whatever you have in your head. For those people who are not too familiar with these kind of sequencing technologies, just want to give you like a very brief overview. So the first generation is the classic Sanger sequencing. So even if people say like that's kind of the old methodology, it still is used in molecular biology labs and still has this reason due to certain advantages. Then majorly what was behind the release of the bird genomes was the second generation sequencing. And here we're talking about short read sequencing, something like you probably heard about the Illumina sequencing. And then there are also newer technologies, third generation sequencing, which are nanopore and PacBio, which become increasingly important. There's also fourth generation, but I don't want to go into detail for now. So basically, NGS was the key. But really, the thing is, you want to ask, so what? So we have now all these genomes, probably we can generate more and more, and there are these huge consortia that generate all these genomes. But what can we actually do with these? Just accumulating data is probably not enough. And there's like two key questions I personally would like to ask. So the first one is, what does it actually mean to have a genome sequenced? So obviously what you know is, if you look around, your relatives or friends, they're all human, but still they're different from you, and also their genome differs. So is there really the human genome? No, there isn't. Every individual is different. So what does it actually mean to have, for example, the human genome? And the other question is, and obviously the more intriguing and more important one, what kind of approaches can we use to answer, to tackle important biological questions using these huge data resources, for example, these genomic sequences? And what I want to do today is I want to give you 3 examples of my own research that actually link to these questions and that link also to the flock of genomes. So the first question I would like to address is, what does it mean to have a genome sequenced? So there's a relatively nice News and Views article asking the question, how complete are these genomes? And in avians, there are certain problems. So one of them is highlighted by the group of Alexander Su, who's interested in transposons and also in repeats in sequences that associate with that. And these regions, they can be very, very poorly resolved using these classical approaches. So that's one problem with the avian kind of genomics. The second problem is that it isn't highlighted here, is that unlike human or mammalian genomes, the avian genomes have a huge heterogeneity in the base composition. So in particular, the GC content in certain regions is really high. So you have so-called GC-rich regions. Again, something NGS has a lot of problems with. And there's another issue with the birds. The karyotypes are really stable, and that means if you have problems in one bird species to sequence a certain region, you also have problems in many other species, leading to the fact that apparently many genes were supposedly missing in birds, which are not. They're just lying in these GC-rich regions. So there's also problems that are more pronounced in birds relative to other genes. So again, what does it mean to have a genome sequence? I actually do know that because I've been involved in one of these huge genome consortia projects. So at the early days, it was really easy to, not easy, but it was relatively easy to get a high impact publication with just the genome and a little analysis. Nowadays, because there's so many genomes available, obviously it depends on, you know, what's your story, what kind of data you have, and you have to be yeah, also a little bit lucky. But really the thing is that these genome consortia, although they're so common, they're still very, very hard. So I can only advise you, for those who are asked to be involved in, for example, genome assembly or something like this, really, really think hard, because this is a big effort. So I really actually do know what it means to have a genome sequence, and also how complete this genome is. So we were involved, or I was involved in the release of the great tit genome. That's a passerine bird. It's abundant in Europe and Asia, and it's a modeled species for ecological research. And we released the genome in 2016 in Nature Communications. And we actually got intrigued by all these facts, all these issues I just mentioned, that these genomes are not really complete. So we went out for a hunt together with my colleague and collaborator Veronica Lane, and we started to explore information of the sequence genome that we disregard, or that other people throw away. And these are the so-called unmapped DNA and RNA reads from our great tit genome. So just to give you a little overview for those people who are not too familiar with this kind of genome sequencing or resequencing. So what usually happens is, if you start to have your genome, you really cut it in teeny tiny pieces. And these are called raw reads, depending on your sequencer. They may be 50, 100, or 200 base pairs, sometimes longer. With a third generation sequencing, they're longer. And then what you can do, you can actually use these raw reads and try to kind of assemble them together. Or if you have already a genome from a related individual, then what you can do is you can actually map it to this genome, just put it on, and then you kind of look for variation. But what people don't know, or what is usually not mentioned so prominently, is that there's many information that goes kind of in the bin. And these are so-called unmapped reads. Some of them go in the bin because they are of poor quality, or some of them, they go into the bin because there's nothing to map them to. So for example, there could be genuinely new sequence information in the individual you have, but you don't account it because not in the reference genome, so you try to kind of throw it away. But it could also have other information. So as an individual, it's always some kind of metagenome. So for example, it has bacterial species in it, or other species. So all these kind of host DNA also would go into the unmapped reads. And something similar applies to the RNA-seq data. So that's for RNA. Again, you're dependent on your gene prediction. If your gene is not predicted, if you have a missing model, you might have the gene in there, but you wouldn't identify it because, you know, it's kind of missing in your reference. Or something also common really for birds is that your gene models are incomplete. So you have only partial genes, and then that leads to a downstream analysis that maybe genes are shorter in birds than in other species, which is not the case. It's just because these gene models are incomplete. So we did this kind of unmapped read hunting from our reference genome. So we had RNA-seq data, we had bisulfite data, we had genome data, and we were actually digging in the trash. And I don't want to bore you too much with the technical details, just want to go to the summary, what actually came out of this, and also give you an idea of, if you have your own genome or you want to do your own kind of analysis, it's actually quite interesting to dig in the trash. So the first one is that we were able to reconstruct many missing genes. So something I told you before is that apparently there's been papers about it, you know, birds have less genes because they need to fly and they need to be lighter and something like this. Most of these actually missing genes, they do exist. Other publications have shown that we were able to reconstruct many of these. The second thing we found out was a huge contamination in one of our RNA samples. It was actually contaminated with Arabidopsis. And because we don't work with Arabidopsis, the source of this contamination actually came from the sequencing facility. And again, if you dig a little bit in the literature, you will find that this is not uncommon, that apparently trees growing next to your sequencing facilities may contaminate your samples. And it's always good to really do check that. So what actually happened is, after we found out we got some free runs from the sequencing center because they were embarrassed. The third thing we found out that there was a deposited reference mitochondrial genome of our species, Perus major, but we figured out that actually this deposited reference sequence was wrong. It belonged to Perus minor, so another sister species. So we could fix that. Also, obviously, other genomic data like the mitochondrial genome is also abundant when you do DNA sequencing. And then when we figured out, this is just one example, that many of the unmapped reads belong to Plasmodium relictum. So as I told you, that's a parasite. So some form of avian malaria our bird was suffering. There were also other pathogens that we identified, and that's something that is not uncommon. So it's actually quite a good way to identify also pathogens, maybe something that may be even interesting for kind of some sort of a, virus analysis if you want in our times, very important. Okay, so that concludes the first part. The second part goes a little bit more, how complete is the genome? Asks actually another question. So if you're not aware of this story, and I'm very happy that I was playing some part, I didn't play the major part, but I did play some part in the story. But that's really something, if you want to consider jaw-dropping science, it's a jaw-dropping result. So I think if you haven't heard about this, that might be something you want to listen to. So what really is identified is that in songbirds, so in a large proportion of birds, there is actually an extra chromosome that only occurs in the germline. So it's not in the somatic tissue. So usually when you start to sequence your bird, you take blood. So birds have a nucleus in their blood cells, in their red blood cells, so you can extract quite nicely DNA from it. And then usually that is used to sequence. And that's obviously somatic tissue. So all these birds that have been in a flock of genomes, they have been using only blood, so they've missed the germline chromosome. So you would say, like, what does it mean? Not all, there's an extra chromosome. So this is actually known. So you might even know this from school. So not all cells have the same genome. We know that there's sort of disease karyotype variation. One of them is Down syndrome, would be one example. So that's also something from human context. And there are also other examples from other species, like ciliates, lampreys, nematodes, and various other eukaryotes. What happens is that during the germline soma differentiation, there's a change in how the genome is organized. And in our particular case, it's basically a degradation of one chromosome. So how does this look like? I have to adjust a little bit to zoom here. So if you see my mouse pointer here, that would be a somatic cell, that would be a germline cell, and you have a particular antibody that would actually highlight this. So you see here in the kind of germline cell, you see that there's this extra chromosome, and you can also see that's huge. So we're talking actually the biggest chromosome of the zebra finch in this particular case. Yeah? So that's not great tit. Great tit is also somewhere, has also this GRC, but this particular thing was found in zebra finch. So when we did the story, or Alexander Su, who's the main guy behind this analysis, there apparently was a publication where they reported the GRC, which in some ways was a little bit disappointing because we were very close to release this, and maybe kind of led to the fact that it didn't end up in the most high impact journal you could think of, but still I think very interesting story. So they basically reported one gene that they could identify on this particular GRC, on this germline restricted chromosome. So they conducted some sort of subtractive genomic pipeline to uncover single genes. So what you have to understand is that these genes that they identified, or this particular gene, has a copy in the, let's say, autosomal genome. So there's basically a copy that differs by a few different positions, and it's located on the GRC. So that was the story from before. It was published a year before us in the Current Biology. But Alexander and his collaborators found many other genes lying on the GRC, hundreds of genes. So this was then the part when I came in, because just by chance, we actually had a data set that contained proteomic data, unbiased proteomic data of reproductive tissue of zebra finches. And I told Alex, yeah, well, it's actually quite straightforward for us to investigate whether we're not only finding something on the RNA level, but to also find something on the protein level that would support that these genes are actually expressed. Because it still is a possibility that this genome actually is just this extra chromosome is some sort of parasitic chromosome, which also exists in their examples for these kind of phenomena. So we were really asking, is the GRC actually translated? Second question was, is it actually possible on a genomic, on a proteomic level to distinguish these GRCs? Julia, I hear some sounds. I don't know who that is. Okay, it's silenced again. So can we distinguish these GRC-specific variants? So we can distinguish them on RNA level, but can we distinguish them on a protein level? And how conserved are these GRC-specific variants across songbirds? So as I told you, luckily I was in a position of being able to provide proteomic data that's been conducted in Sheffield, as Julia mentioned before. So we were able to extract 7 testes and 2 ovaries. So you think like, why 7 testes and 2 ovaries? So that was actually the fault of FedEx, who lost the first parcel shipped to Singapore. So this is basically our backup set. So it was a little bit unbiased, a little bit biased, unfortunately. And then my collaborator in Singapore, Dennis Kabai, prepared these samples and did a mass spectrometry analysis, and the proteomic data analysis then was done by my PhD student in Sheffield. So what do you have to know about these kind of proteomics? So it's a little bit similar actually to NGS, to next generation sequencing. So you cut your proteins into relatively short bits. These are cut with specific enzymes, and you get very tiny peptides. Unfortunately, it works like this, that not the whole protein is cut perfectly and that you have all protein covered. So there's also some sort of luck component. And you also know that usually the cutting happens at an arginine protein. And then what you can do is you basically have 2 versions. So in this particular case, it's one gene that has one version on the reference genome and one version on what we suspect is the GRC copy. So these proteins are really, really similar, but as you can see, there's one peptide that actually supports the one position that is different between the reference and the alternative allele, and that's this D in this particular case. The version on the GRC is supported. And very similar is basically the more mutations you have, the more amino acids are different, the stronger you support it. So in this particular case, you actually find a peptide that supports 2 sites within the peptide. And that gives you an idea of whether the protein is translated or not. So we were able to identify 5 genes that actually did show GRC-specific translation. One big thing was, what we know is that the GRC is inherited through the females. So one of the hypotheses was that it actually only plays a role in females. It's not translated in the males, but we could really show that we also find translation of these specific genes specific to testis tissue. Then also the alpha SNP, which in our case we call NAPA, so that was the gene that was published in Current Biology, came also up in our study. There was actually a big relief, or it was like, you know, an additional check that we found, yeah, cool, this gene is there, and we find actually 4 more that actually show expression. And that were the 2 key messages that apparently, yes, there is translation. We can pinpoint this for 5 genes. There are many other genes that show expression or translation, but we're not able to distinguish them. That's basically the problem. So there could be more genes, but it's just like with the kind of peptide composition, we are not able to do that. So then an additional kind of analysis I conducted, basically Alexander was interested in how old this GRC might be. So he's actually able to pinpoint different genes on the GRC on the phylogeny. For example, that if the gene is very old, then you can actually map it back to a very old phylostrata if it's very recent, then it's a very current phylostrata. So many genes are really young, and that's a little bit the hypothesis we have, that there's a constant turnover of genes into a GRC with many copies being kicked out again, but some of them actually gaining a function, or that's at least the hypothesis. So what I conducted was the NGS analysis, which is basically a test of how conserved on the DNA level the protein coding DNA is. So if you would have only random mutations, you would expect that this ratio is close to 1. But what we could show is that for many genes, for in this particular case 8 genes, mainly these old ones, this DNDS ratio was much lower than 1, illustrating that there are some sort of purifying selection. So they probably do have indeed a function. So that was kind of wrapping up the GRC story. So what we could show is that the GRC is not only present, but it's also translated in zebra finch. We could distinguish specific variants from homologous regions of the autosomes. These GRC-specific variants, they're conserved across a number of all passerines. So some of these genes are really old. And we could actually also show that there's purifying selection acting on that. Again, noting or hinting at the notion that GRC-specific genes actually do have a function. But we also know, I didn't show this because there was actually work from Alex. Alex was wondering, what kind of genes are actually on the GRC. So he did say some sort of, gene ontology enrichment analysis, and these being highly significantly associated with reproduction. So the homologous versions of these are, involved in a reproduction. Okay, so that was the story about, that was the first question, these two examples. How complete is a genome, or what does it mean to have a complete genome? So you probably never can have a complete genome, but I think too interesting, and especially the GRC, so really stunning story about, what a complete genome actually means, and kind of, speaking against this idea that all cells have the same genome, which in broad is correct, but in this particular case, it's not. But this is obviously very specific to particular species here, and a question that also arises from, if we have so many bird genomes available, can we actually use these in some sort of, large-scale big data kind of approach that goes maybe beyond genome-wide association studies or something like that, but really trying to take advantage of these huge comparative resources. And that ended up in Genome Research, was published this year. Mainly work from these 3 students of mine, Liban, Matthew, and Joe, who were master students or PhD students in my lab. So what we wanted to do is we wanted to actually get an idea of how can we associate phenotypes to the genomic regions that are basically driving their diversity. And I think we probably all agree that the Beak, the Bill Beak, is maybe a model phenotype for that, because it's incredibly diverse. It really well captures species diversity, and it is one of these big examples of the targets of natural selection. When we think, for example, about Darwin's finches, that have been part of this big origin of species. So then the first question would be, okay, we have these genomes and we're interested in the model trait. So how can we actually how can we actually measure this model trait? So when you think about the Beak, I'm obviously not someone who's was the first thinking about this. People have done this for a long time. Scientists want to measure things, so they can use simple methods like length, width, and depth. But my collaborators in Sheffield, they actually went one step further, and they did 3D morphometric scans. And the idea behind this is actually very well described on this website. It's citizen science project where you can log in, get a medal, start to geomark your birds. Basically, the idea is not only having these simple kind of measurements, but also capture curvature, like, for example, in this particular example. So what happens is that usually a single bird scan is kind of mapped by 4 individuals on this website. Then the moderator or the guy who's organizing this basically compares these 4 different landmarks, or in this case, phenomarks. And if there's a disagreement, he would manually check. If it's not, then he says, okay, that's relatively accurate. And then what you can do is you can do some sort of phylogenetic trait reconstruction. This is what my collaborators of Mark My Bird, so that's Gavin Thomas and Chris Cooney, basically did. That was a Nature publication in 2017, where more than 2,000 species actually were evaluated. So how did we start the project? So we were basically interested in what genes could be potentially involved in beak shape evolution. So many species have done it. There's many scientists have done this. There's a few key publications. So for the connoisseurs of beak shape evolution, key genes have been identified: ALX1, CALM1, BMP4. So there were a number of candidate genes that have been identified. If I modify these in some way, a chicken, for example, has a very modified beak, when it's born. But what this kind of analysis also showed is that the genetic architecture of beak shape probably is not very simple. It's likely to be relatively complex and probably not so surprising. Some important genes showed large effect sizes, probably the reason why they were identified. They were also deducted that they have been experienced strong detectable selection pressures. But it really seems that different bird lineages have different genes underlying the diversification. And so you couldn't repeat a finding from one species in another one. So that actually led me to think, well, these species-specific candidate genes, are they actually important over broader evolutionary timescales? So that was the big question I wanted to address. So yes, they play a role in a single species, but what about on the macroevolutionary scale? So what we did is we used the genomic data that included the flock of genomes, but also additional data that were released in the meanwhile. We had 72 bird genomes from NCBI, and 57 of these were annotated by RefSeq. So that's the annotation pipeline from NCBI, which means that the genome is of higher quality, and you have also some information about the genes. So we went for two approaches. The reason is we were not only interested in genes that might be involved, but we were also interested in non-coding regions. So we constructed whole genome alignments. So these are 72 bird genomes, and we're talking really diverse set of species. That's a very big last set alignment. We also did gene alignments using pranks. So this is based on this 57 annotated birds. Again, it's a very big data set. So this is still it's on a scale that I think only a few labs can actually do on using so much biological information at the same time. To actually narrow down our genomic information, our non-coding regions, we used information from chicken, where so-called avian-specific highly conserved elements were identified. That's a publication, Nature Communications 2017. So we were basically able to get quality alignments of 230,000 genomic regions and approximately 10,000 genes, and we used PAML to calculate substitution rates for genes and for non-coding regions. So that's the tree I showed you earlier from Cooney et al. 2017, more than 2,000 birds. The figure can be read like this. So if you have, for example, birds that show a blue color, a relatively cold color, then their change of beak over evolutionary time is relatively slow, while warmer colors would indicate a fast change. So for example, here you have the passerines, and the passerines, the beaks change quite drastically. So as I told you, so Mark My Bird published more than 2,000 species. We don't have information of 2,000 species. So really the problem is to prune the tree down to the information we have. So we have something like 72 species or 57 for the gene elements. And what you have to understand, or what's important, this is not like just having phenotypic information for these number of species. You still need to have the phenotypic information of these 2,000 species, and then you can prune it down. But the information of the other species is still hidden in here, although you don't have genomic information of the actual tips of the tree. So we can only do the analysis for these kind of species only. So how can we then combine the morphometric and the genomic data? Well, we went for actually a relatively simple approach. We started to bin our tree. In theory, these kind of substitution rate models, they can calculate in a kind of most free model. They can calculate for every branch a change of substitution rate, but actually the model becomes computationally very intensive, and also you have more than twice the number of species as branches in the tree. So that means you would have, with n parameters, 2 times n estimates, and then obviously that it's an over parameterization. So we started to bin these, and what we did is we obtained basically bins. For example, if we have little change of beak shape, and then it would get a lower number, for example, this. But if the beak shape change is very dramatic, so for example here and this one, then that would get a higher number. And those branches that show similar rates of beak shape change, so the morphological change, they would be binned in the same category. And then what we can calculate is the rate of substitution change, so the genomic change. And in a very ideal world, we would expect some sort of positive correlation. So where there's a lot of morphological change, we would also expect a lot of change in, for example, on the protein coding level, something like this. This does not necessarily mean that there is positive selection. You could also think simply a model that incorporates only purifying selection. You still would have a thing. So the beak shape change basically goes up when there's a relaxation of selection. And even if the whole model doesn't involve any selection, so it would be simply neutral, so simply due to changes, for example, in population sizes, or simply due to random changes, you would still expect that higher morphological change would also result in higher genomic change. And that's a very simplistic model, and I agree you can make up something much more complicated. Obviously that's possible, but this is some sort of null model we assumed. So again, I want to walk you through the results relatively briefly. We addressed protein coding genes because that was where the candidate genes came from. So we didn't find any significant correlation of the NGS or something I've just explained to you and the rate of beak shape. What was very stunning is that we had more than 10,000 genes, but only 1,400 genes actually showed significant variation in the NGS across group branches. So that means that all the other genes, they're actually very, very conserved, and they wouldn't be the candidate genes you would think of if you say like, this explains all the diversity of beaks we have, because they're simply on a genomic level too invariant. And what was really the big thing, and actually very disappointing for the first master student working on this, all these candidate genes, they were genetically invariant. So the candidate genes have been identified, they cannot explain what's going on the macroevolutionary scale, simply too conserved. They're still involved, but not they're not drivers of the macroevolutionary diversification. Then we went to the non-coding regions. So again, we identified something like 40,000 that showed significant variation in their substitution rates, a little bit similar to the DNDS variation. And then what we did is we tried to identify genes that are nearby these particular regions that had enough variation to explain this morphological diversity in beaks. And we actually came down to 163 genes that were not only near these regions, but that also showed this significant correlation I was talking about before. And these 163 genes, they showed a very strong overlap with a gene set derived from mice that were included manually curated craniofacial genes, 511. Published in 2014. And I think that was basically the turning point of the whole thing, because we realized, oh, this gene set we have actually seems to be really, really meaningful, because at the end of the day, we only had the genomes and we had this huge data set of morphological change, but whether this approach works or it's only like a nice idea wasn't really clear until the moment we identified these genes. We also took into consideration that maybe these non-coding regions not acting only in cis, but could also act in trans, so far away, have maybe a target gene that's far away from the actual genomic region. We identified 144 potential binding proteins, but among these were, for example, GSC and SMART. Again, for the connoisseurs of the beak shape evolution, these are two of these candidate genes that I said that are actually invariant. but maybe regulators, genomic regions that regulate these genes, they might be involved in this. So that, again, was another hint. So to play this a little bit more down, I started to investigate these 163 genes a little bit more carefully and started to do a phenotype enrichment analysis. So basically asking whether there's any human phenotypes enriched in these 163 genes. And I got this list, and you probably realized already that I'm not a native English speaker. So all these words or terms, they didn't tell me anything. So I started to look them up, and then suddenly realized, oh my God, it's a huge chunk of these is related to craniofacial features. So for example, the smooth philtrum is basically that region here. The vermilion border is basically this part here of the lip. But we also had things like macrotia, as you can see here, or a large face, or hypotelorism. So that means basically the distance between the eyes really low, small. And again, this is not some cherry pick data set. Was the first thing that came up, and I think, and it was pretty constant depending on what kind of thresholds I'm using. Sometimes other terms came up, but all within this kind of phenotypes that I think were for me very surprising and actually showing me that this kind of method seems to have very good potential to, at least on a large scale, on like complex phenotypes, shed a little bit more light into what are the genomic underpinnings of these. Another thing that also came across was I was interested whether there's any pathways that are actually involved in or associated with these kind of genes. And something I realized is, as I told you, even for the protein coding genes, we didn't find any that showed this positive correlation, but we still identified some genes that were invariant, or that showed at least some sort of variation that they might explain and might be associated. So we looked a little bit deeper, and that what came up is that for the non-coding and for the coding kind of tests, something called the ESC pluripotency pathway was involved. And again, because I'm not an expert of these kind of things, I started to look a little bit more detail, and then suddenly realized, this is actually something that involves the really classic pathways, BMP, Wnt, RAS-ERK pathway. So these are really the classic pathways that have been shown in the Beak shape literature, Beak morphological literature, as being associated. And one of the key findings I found really interesting is, for example, for the BMP, there's BMP4 here. We didn't identify BMP4 in our study, but what we did identify was actually genes. So for example, these SMADs here I showed you before, that are involved downstream of BMP4. So of course, if you modify BMP4 in your chicken, for example, then you also modify the downstream players that we think are on a macroevolutionary scale associated with the diversification of peaks. Again, really nice in line with what we think our gene set actually means. So just to kind of wrap this up. So I told you about this idea of this positive correlation. So I want to go back to this. Okay, yes, it's a really simple model. But although we didn't identify any protein coding genes, doesn't mean that they're not involved. So it could well be that because we had only 50 plus species for the protein coding set, it could well be if we have more species, we gain much more power, and maybe we're more, there's more possibility to find associations. Maybe my hypothesis is just stupid. Maybe we need to think about something more smart or more complex, which I think then makes it actually quite difficult to analyze. But still, this is possible. And then there's also the saying that maybe it's not the protein coding genes that are the key players, but it's actually the non-coding regions. And this is something we went for in the paper at the end. Yeah. So now I was talking all about this, they are important on a macroevolutionary scale, and these are the genes that are identified, and still it's just a gene list and showing some gene ontology enrichments. So really the question is, if they play a role on the macroevolutionary scale, you still should see some evidence if you look in a particular species. So in a particular bird species. And we're lucky enough because, as I told you, birds are some key models for the role of selection, that actually people did sequence a large bunch of birds that were differing in the beak morphology. And this is the classic Darwin's finches I was talking about. And what happened here is that there are two groups of Darwin finches. Some of them have a pointy beak and some of them have blunt beaks. And this is obviously a very nice kind of possibility to ask whether genomic regions that we think are important for the macroevolution scale, whether they actually differ between the pointy species and the blunt species. So these are actually not species. These are two species or three species for the pointy ones and some subspecies for the blunt beaks. And we were basically asking whether we could show some sort of difference. And a very cool measurement for that is the FST statistic, which basically gives you some sort of level of differentiation between two groups of individuals. So in blue, we have basically the regions we identified in our data set, and in green is genomic control regions, because as I told you, these are highly conserved non-coding regions. So what you expect for them is that obviously they have very little diversity. FST is highly affected by the genetic diversity, so you would expect a drop here. But really what is important is that it's still a huge discrepancy between our identified region and a genomic control, meaning that our regions actually indeed show higher differentiation in the Darwin's finches compared to a random model. And that, again, further supporting the idea that these non-coding regions we identified, they are probably, they actually also play a role in a microevolutionary scale, and you still can show this in this kind of data set, which really makes the story round and yeah, ends up a very nice result. So the conclusions about this is, yes, we can use these massive data sets from the flock of genomes and all these other stuff that is coming out soon. So for example, BERT 10K, there's vertebrate genomes, there's the BAT 1K genome. Obviously more genomic data would be helpful. They will be coming and we don't need to wait for too long. We found actually very limited evidence, at least on a protein coding level, for positive selection. So that's something really, one of these ideas is Darwin right. One of the examples was that, oh, this must have been adaptive process, how these peaks changed. So here we didn't find any evidence, but I told you there's these technical limitations that probably need to investigate further. But if I had €50, I would basically not bet on the genes, I would bet on the non-coding regions. Obviously, our methods to identify positive selection in non-coding regions, especially in a comparative kind of way, are still relatively limited. So we need also to develop some methods in this respect. So to sum up my stories I showed you three stories. First thing is, if you do next generation sequencing or any type of sequencing, check your sequences. You might want to use alternative approaches. Don't just go for the standards. There's always something interesting around the corner. Think out of the box. Maybe that doesn't only apply for genomes. You know, yes, it's good to have an idea. You know, set a hypothesis, test a hypothesis, it works out. I know that many PhD students, for example, get very frustrated, you know, if the experiments don't work. But that's also the charm and the exciting bit of science. Sometimes, you know, things come up you didn't expect or you couldn't even imagine before. And I think this GRC is one of these stories. And yeah, linking phenotypic and genomic data over macroevolution times is actually possible. One approach I've shown you. And I think that's maybe something that people have forgotten a little bit about in the last 10 years, but I think it will become increasingly important, especially, you know, once we have all these genomes released. So just to wrap up, talk a little bit about me. So Julia introduced me quite nicely before. I'm not only doing avian genomics, although yes, my recent work has been quite surrounded on avian genomics. I've been also involved in the Alpine Genome project. There's also been other work that goes more into the molecular evolution part, disease hotspot, but I'm also interested in population genetic theory, for example, time series or microbiome evolution. These are all things that are really brand new, and people just, you know, start to think about, you know, how does the microbiome actually evolve? So at the moment, people usually just observe and describe. And something that I've been, through collaborators, quite interesting is the process of aging, and here in particular NAD metabolism is one of my key players. But in the next 5 years, as Julia already described, one of my, well, probably the prominent research that we're doing will be surrounded on epigenetics and mesothelium evolution, because I've just been awarded an ERC standing grant. So why epigenetics? Well, I told you all about this genetics, and now I want to go already make a step further to the epigenetics. Well, the thing is, one of the conundrums is that this positive selection with this Darwinian evolution is really on a genomic level not easy to show. And one of the possible solutions to this is that actually it's not a genetic variation, but it's the epigenetic variation Darwinian evolution acts on. It's quite nice, and many people do work in plants on this. But the problem in animals is that they have a separated germline. So that means that most of these epimarks are not heritable. So even they have an effect, and maybe selection can act for like a generation on it, it's not given to the next generation, so it wouldn't really fulfill the criteria Darwin set out. Nonetheless, I still want to decipher the footprints of epigenetic-driven adaptation. The reason for this is that first of all, we have a new method for this. I want to use DNA methylation. That's a very well understood epimark, but I really want to look at the genetic anchors of this epimark. And these are obviously genetic elements, and these genetic elements can be exposed to natural selection in a kind of classic framework. And I want to use an avian system for this. Obviously, I've used to work, or I did a lot of work on avian system. But really what I think is such a great system, because DNA methylation in birds works very similar to humans, but on the other hand, they do have an external embryo. So if you want to do epigenetics, you need to think about early development, because the reason is that actually epigenetics, yes, there's an epigenetic reprogramming really early in life. And that means if you were interested in changes that are actually inherited, you need to look at these epimarks during embryo development that are stably methylated. And these are is really only a tiny fraction, because there's almost like a deletion of all these epimarks and then a reprogramming. It's not really understood how this is maintained. And yeah, that's one of the key parts of my ERC. Okay, and yeah, why this is timely? Well, it combines genomics and epigenomics, and what we know is, for example, in cancer, is there's a functional, very important role of epimarks. Interesting thing is also what people forget is that these CpG sites, so these are these sites in the DNA that are methylated, they are highly mutagenic, and apparently, if you take, let's say, standard cancer tissue and just screen for mutations, 50% of the mutations are at these CpG sites, but on the other hand, CpG sites are heavily deprived in the genome because they're basically kept at low frequency. Part of this is the mutation equilibrium, but it's simply because, of their functional implications. Yeah, and what I can do now is to use nanopore sequencing. It's a new novel sequencing approach that can not only do genomic resolution, but can also do epigenomic resolution. So you can actually obtain signals of DNA modifications through nanopore sequencing. And that gives us some idea of how I later on could actually start to develop experimental tools to look a little bit more in detail. And what I think is really sexy and attractive about this is that it's possible to transfer knowledge from the relatively boring birds to human disease and beyond, and also get some general idea how epigenetic works in vertebrate system. Okay, then I want to thank my students and collaborators about these three projects I presented to you, and I'm happy to take questions.